Cheers, just as I'm drinking a cup of tea. Okay, let's see how we go this time around. I just had to abandon my year 12 class because the internet was all shoddy in that other room. I had a meeting on Zoom like immediately before. In fact, I was 2 minutes late for my live stream because I was in that meeting. And then I switched to the Zoom room to broadcast to you guys, and the internet just went to the dogs. Hi, Rehab. Okay, so we're going to do some gene technology stuff today. So isolating target genes, inserting target genes. I'll show you kind of where in the spec it is. It's topic 8 for EDD Excel. They don't make too big of a deal of it in the EDD Excel spec. Basically, you need to know, and we're going to discuss before we get too stuck into any of the details, what is the purpose of gene technology? So we're going to discuss these bits and pieces and look at the mechanisms by which it's done. I didn't import any questions onto here because they wouldn't download. Actually, I might be able to do that. It's probably downloaded by now. Let me just check before I start sharing my screen. And let's have a look. What do you mean tap to download? I have already downloaded. Maybe the download. Oh no. Well, we look like maybe we've got the EDD Excel document, so it's a pretty giant PDF. So we'll see if it actually is going to load. Oh, it's just a notes template. Yeah, I don't know. I'm like working between two different computers right now. I'm tapping on a document that is the document that I want, and it's opening up a PDF of a document that I don't want. I'll just pull some questions off a screenshot and then move it across. Let me know if there's any sort of glitchiness in terms of the video. In fact, so would you be interested? I can maybe start by showing you the spec, and you can see how each of your exam boards specifically phrase it. Okay, let's do that. I am back in the UK. I made it just about by the skin of my teeth. So I'm now, well, I wouldn't say I'm back on UK time zone. I am in the UK time zone. My body clock is still a little bit, needs a little bit of adjusting. Edith, your teacher refused to set any work. Okay, I'm going to dive into the class and I'll save questions for the end. So it'll be most useful for people watching the recording. Okay, so I'm going to share my iPad screen and just sort of recap what's on the spec for each of you. Because you'll see how different exam boards phrase it in different ways. This is the AQA spec here. I'll just use my pointer. So it's 3.8.4.1 essentially, and it takes us through basically creating DNA fragments, use of restriction enzymes and marker genes to insert the useful genes. So it actually breaks it down into quite a lot of detail. This includes some of the stuff on PCR and in vitro versus in vivo gene cloning. The OCR spec over here is, again, we've got DNA profiling. Well, we won't cover that today. I might come back to that later on. We've got PCR and stuff like that, but it's the principles of genetic engineering. So isolation of genes from one organism and placing the genes into another organism and using suitable vectors. And then techniques, restriction enzymes, plasmids, DNA ligase to create recombinant DNA. So this is basically the area that I'm going to be focusing on for today's session. EDD Excel is super vague. Know how drugs can be produced using genetically modified organisms, plants, animals, and microorganisms. Essentially, that is the part of the spec that covers this. They don't go into as much detail. They probably won't require quite as much detail. I will maybe look for some EDD Excel questions, so to exemplify to EDD Excel students what level of detail you are required to know. So, okay, just trying to keep, I'm going to just pull out extract the YouTube comments so I can keep them visible, hopefully. And then move a few windows around. So again, you're going to need 4 color pens, red, blue, green, black for the color taking system. I'd love to do a quick Q&A with you guys at the end, actually, just to find out about what your teachers have been setting, and maybe ways in which TT can help in support that, essentially. Okay, let's go back. Pens and paper at the ready. If anyone watching this wants to put some timestamps underneath in the comments, that would be dope. Okay, so let's switch over here. So I don't want to start with inserting. Before you insert, we're going to isolate. In fact, let's begin with a very brief conversation. Why would you want to cut a gene out from one organism and add it into another organism? Like, what are the general purposes of that? Let's just go over that in the chat just quickly. Why do it? Some potential uses. What DNA? There's 3 words we can use to describe DNA. And one of them is universal. So the mechanism that allows us to do this is because the code for a gene is the same as it is in bacteria, as it is in fungi, as it is in plants, as it is in humans, in mammals. Okay, so for example, insulin production. So to produce, what is insulin? What do we want it to make? Desired characteristic. Again, we can be more specific than that as A-level biology students. To express a certain gene. Golden rice. Again, what are we making? What are all these things making? Variation is an interesting one. I'll come back to variation. Let's just, golden rice is rice that has been genetically engineered to produce a certain, in fact, I think it's actually a vitamin, isn't it? Golden rice. Proteins. Okay, so basically, what is a gene? You're adding a gene. What is a gene? A gene is a sequence of DNA that codes for a specific protein. So again, desirable characteristic. The characteristic is brought about by the protein. So basically, it's this organism makes this awesome protein. Super useful. That organism, it would be kind of useful if that organism could do this thing. So let's insert it there. So this could be for herbicide resistance. It might be you want to add herbicide resistance to a crop so that you can then spray the whole field with that herbicide, and then it will kill all the plants, all the things that are going to compete with it, reduce competition, except the genetically modified crop. It might be a pesticide. You might want to take a pesticide producing a protein and insert that into the plant. So the plant makes its own pesticide. So the insects are less likely to consume it. These are all the sort of theoretical reasons behind why you might want to do it. You might want to make a human protein such as insulin. We need it as a therapeutic, as a drug. I think it's probably fair to call it a drug, as a therapeutic. And so we want to make lots of copies of this protein because some humans don't make it very much or at all. And therefore, by having large quantities of human insulin, there's not going to be sort of reactions to it. We used to use pig insulin and other types of insulin. It is vitamin A. Thank you, Matilda. So yeah, you might just want to add an additional. So again, vitamin A itself is not a protein, but it's probably a protein, maybe an enzyme that makes vitamin A. If anyone can tell me a bit more about golden rice, then let me know in the chat. Variation, interesting. What is the general result on variation, genetic variation, of using gene technology? I'm back in the UK. Is the answer to your question, Russell? Generally speaking, more or less variation, genetic variation. You could say at the individual level, the recombinant organism, the transformed organism that you produce, has a novel set of DNA. Technically, that organism, you've got some slightly increased genetic diversity. You've got a new combination of genes that never existed before. There's tiny bit of increase there. But what happens if we then make that crop species herbicide resistant? What are you going to do with that new genetically modified organism? You're going to grow it, and you're going to grow it en masse in monoculture because no other plant has that gene. They're the only plants that have that gene. So you're just going to grow thousands and thousands or millions of those plants with the same genome, genetically identical, all produced probably through vegetative propagation if you're an OCR student, to to reproduce as many of those things as you can. So overall, over time, you end up with a decrease in genetic diversity because you make the one really useful organism and then you make clones of it. And by then growing a monoculture of clones, you end up with a reduced genetic diversity in general. Yeah, the gene pool. I mean, obviously, that's why it's really important in agriculture to preserve wild types. So like the old sort of slightly lower yielding varieties, because what's the danger of having less genetic diversity? Or you can flip it, what's the benefit of having a diverse genetic diversity? Especially in agriculture, but it goes for any walk, any species, any population. Okay, so anything with a smaller genetic diversity, it's a bit, think of your genetic diversity as like a Swiss Army knife. The more diversity you have, the more tools you have on your Swiss Army knife. So the more you are able to cope with changes, generally speaking, that's going to be environmental changes. If you've got a huge selection of tools to choose from, if you have a drought year, you've probably got some old drought resistant genes back in the old back catalog somewhere. If you've got a really wet year, you've maybe got some wet resistant genes to deal with. Maybe you get more fungus infections, and so you get some resistance to that fungus infection. So if you've only got one very narrow genetic diversity, you are much less able to cope with change. One disease might wipe you all out, especially if you're genetically identical. Okay. So let's run through the mechanism of chopping out target genes. So the target gene is the one that we're going to find useful. It could be the insulin, it could be the herbicide resistance, whatever it is, we want to get the useful gene on it. There are 3 ways that AQA students need to know how to do this. I have seen OCR questions ask about these 3, but they're generally optional. The main one that you all should be familiar with, how do we chop out a gene? Straight in there, restriction enzyme. You can call them restriction endonucleases. These are enzymes with a specific active site that is complementary to the double-stranded DNA. And you can just say that it's a restriction site. Gene machine is another one. So a gene machine is just basically a sequence. So you know the sequence of the gene that you want. You'll sequence the original version in the organism. You'll be, okay, it goes A, T, T, C, G, T, T. You know the sequence. And then a gene machine is just a machine that will make DNA in the order of bases that you want. So yeah, that's basically going down the roots of the OCR, do more in terms of synthetic biology, but you can just make genes from scratch as long as you know the code. And the third one, this is more related, again, this is not necessarily in your gene technology part of the spec. Coronavirus does this, and HIV does this. What do viruses do? They insert an RNA into your host cell, and they use an enzyme called reverse transcriptase. So that's transcription in reverse, and transcription, what is transcription? Well, it takes DNA and makes messenger RNA. Well, this goes from messenger RNA back into DNA. And so you're doing transcription back, you're going from RNA into DNA. And so the genome of the coronavirus, the genome of HIV, the genome of many viruses, not all of them, is RNA. And it uses an enzyme to convert the RNA into DNA, and that DNA can then insert itself into the nucleus of the cell. And then you can start hijacking the cellular machinery of that cell to go about making proteins. But that's basically, so what's the benefit if we've got the messenger RNA of the insulin gene? What's the benefit of using reverse transcriptase? It does form complementary DNA, cDNA is correct, which does not contain introns. So I'm just going to do Q&A style with you for a little bit here, and we'll go through to some notes. Oh, I've just pulled my power cable out. Everything's going wrong for me today. I'm just having a nightmare. You know, one of those days when you like knock things over and things break, and then you can't reach cables that are stuck behind the table, and get out of it. My cable is stuck in my leg adjustment strap for my table. There we go. My days, you know. Just over it. Just like one of those if it carries on going this badly, I'm just going to go back to bed and catch up on some sleep. Yeah, the camera is going to wobble. It's a wobbly table. It's currently stacked. Do you want to see what my camera's stacked up on right now? This is like homeschool. I've got some Alpro unsweetened almond milk on top of some Waitrose orange juice. Is what's doing my my laptop. Oh my god! Now I've just dropped the power cable again. It's just going to be one of those days. I'm in the kitchen. Yeah, improvised. It's very much. Well, I had to move because the internet wasn't good enough. I've moved as far as I can make out, directly below the router in the house upstairs. Okay, disadvantages of using DNA itself. How many copies of the insulin gene do you have in your cells? Which cells make insulin? Let's just do some rapid fire questioning right here. Beta cells in the islets of Langerhans, in the pancreas. Okay, how many copies of the insulin gene exists in those beta cells, in the nucleus of those beta cells? 2 copies. Yeah, you've got homologous chromosomes. You've got one that you inherited from your mum, one you inherited from your dad, 2 copies of every gene with diploid. So there's only 2 in any given nucleus, 2 of those genes. If that beta cell is banging out insulin, it's just making insulin, it's just producing insulin, how many copies of the messenger RNA might you have in the cytoplasm? Obviously, transcription is going to convert DNA into messenger. For translation, loads. I have loads of messenger RNA. It's just going to be way easier to access. Just there's more of it. Also, when DNA is in the nucleus, it's super tightly supercoiled around histones. It's wrapped up. It's difficult to get at, and it's just hard to manipulate. It's just much easier to work with RNA because it's just more freely available. So that's one of the main reasons why we often use reverse transcriptase. We take the messenger RNA and we'll convert it back into DNA using that enzyme. The disadvantages of that, though, well, it's not necessarily a disadvantage. A difference, though, is that DNA that we make is called complementary DNA, and it does not contain introns. So not that the intron is coding, we don't need that to make the protein, but if you do want to look at differences between different types of insulin, then you'd have less information. Introns are the non-coding parts of DNA that are within a gene that are removed during splicing in transcription. Okay, you're doing a great job of answering each other in the chat. Let's do some more wobbles because I'm going to go back to Zoom over here. Okay, so isolating target genes. Let's choose a blue pen for my title here. No, don't do that. I don't want you to do that. I'm going to say restriction enzymes. I have added most of the template notes to the Exambulance course. So I haven't added these. I'll add these PDFs by the end of the day today, in a couple of hours, actually. Okay, so black pen, real notes. What parts of the DNA do the restriction enzymes use as an active site? This is mostly an AQA spec point, but DNA contains they are palindromic. I'm going to say, yeah, let's go palindromic. It's a nice word. Palindromic sites or sequences. Can anyone give me an example of a palindrome? What does a palindrome mean? No, that's wrong. Never odd or even, it should be. Okay, so if we read it in this direction, it goes NEVER, ODD, OR, EVEN. And if we go in this direction, NEVER, ODD, OR, EVEN. So it's the same forward, reads the same forwards as it does backwards. Hannah, HANNAH, is the same here. So we can say restriction enzymes cut DNA at specific palindromic sites called restriction sites. I'm going to zoom in a little bit here if the iPad's going to let me. Restriction enzymes. Cut, you could say hydrolyze. I'm quite short on space here. Cut DNA. And they're specific palindromic sites. I'm just going to write sites called restriction sites. And these are either side. There's loads of these. They exist pretty regularly throughout the genome. And so they're quite common. They're not that rare. And so you just need to find one. You've got your target gene. In fact, I'll do a drawing in just a second. Restriction sites either side of a target gene. Okay, so let's take our target gene as being some green here. Double-stranded, that's our gene. I'm going to actually draw it. I'm going to have it cut out underneath. So I'm going to use sort of normal DNA in inverted commas in black. So that's either side of the normal DNA, and this might be different lengths. It might not be equidistant either side. And then we've got our recognition site, this palindromic sequence, which I'll draw as a red little section, and then we go back to like normal DNA in inverted commas, and it obviously continues off here. And if we were to look at this and we were to zoom in on this little section here, so we could make it C T T A G in this direction, and then if we're going the same in the opposite direction, C T T A G would be the same in this direction. So this is a little palindromic site here, and the way that it's going to cut, it doesn't just chop straight down like this. It's at the beginning of the sequence and at the end of the sequence. And I suppose if we put some other DNA in here, it would be C, T, A. And I'm just making up these bases. These black bases don't make any difference whatsoever. So we can have and so that would be cutting essentially down here, across here, and down here. And it does the same, assuming that this recognition site that I've drawn on the left of the DNA is the same as the recognition site, as there's multiple different types. So this one is the CTTAAG. There's other recognition sites which would have its own restriction enzyme. Restriction enzymes are specific to the restriction site at which they cut. So this is going to chop out this gene here and here. But the most important thing is that we don't get a clean, like a meat cleaver chop, judo chop. We get in, across, and down at the beginning and at the end of that palindromic site or the recognition site. So if I were to then draw, I'm just drawing the same piece of DNA, I had a shorter bit there, slightly longer bit on this side. And then on this one, we've got the top piece. If we look at, we've got this top piece attached here, and we've got the bottom piece attached here. And I suppose in green I should have our target green is the green bit. So regular DNA is just black. And this doesn't have any dotted lines. This has just been chopped out. It's now a piece of DNA. What can we call these unpaired bases at the end? There's some here, I suppose. Isn't all DNA palindromic? Or if you take the entire length of the entire piece of DNA, no, the sequence doesn't have to be the same. You've got complementary bases, so A is with T and A is with T. But if we look at this random sequence that I've just put in here, this one in this direction, if we were to start here, goes ATT, CTT, AGG, and looking at here with this one should therefore be an A if it was going to be palindromic. But it doesn't do that. So not all DNA is palindromic now. These are called sticky ends. And they are basically unpaired or single-stranded bases. I'm not sure whether that should be an arrow. I think maybe down and across arrow. So they've got unpaired hydrogen bonds on the end there. These are super important because this is without these, that it's very difficult to put the DNA back into something. So let's just come back over here. So when there's a restriction site either side of a target gene, you can basically use a restriction enzyme to cut it out. Should try and keep this in red. And by cutting it out, we can call that isolating it. We are just removing it from its native environment. We can say that using restriction enzyme leaves DNA with sticky ends. I'm going to put sticky ends in red as well because it is a key term. Which are unpaired bases, but I've put that on the other side, so I don't need to add that in there. Okay, so any questions about using a restriction enzyme to chop out a DNA? Again, we've got these different sites. And I don't know if I'm going to leave this on the page or not. What colors can I use? I'm going to use green for my target DNA again. I've got a little bit of target DNA. And obviously the restriction enzyme, the restriction site isn't usually like at the end of the gene. There's usually some length of DNA in here. It might actually be quite a long piece of DNA, but somewhere either side, there's going to be a restriction site. As I said, the restriction site here, I'm going to do in red, but I'll do this one in blue because you've got, I'm going to call this one restriction site B. This one can be restriction site A. So these would require different enzymes to chop these out. But if they're the same, in my first drawing, I use red for both of them, and it's the same enzyme that would chop them out. And it would be the same enzyme in a human, chopping out human DNA, because the site is the same. It would be the same enzyme that would be cutting it out of a plasmid or in a plant. I'm probably just going to delete that bit that I've just drawn there about but basically the restriction site is specific, and the enzyme, the restriction enzyme, is specific to the actual whatever the palindromic sequence is. The sequence of bases is what's going to be the substrate for the enzyme, and so the sequence of bases is complementary. It's not complementary to the active site of the enzyme. What is happening to DNA helix? Yeah, I mean, at this point, one of the quirks of the way that they ask these questions is that they're not going to ask you about DNA unraveling or DNA helicase coming in to make it to like separation from the histones. There's no detail on that there. To be honest, I don't even remember most of that from my university days. And to be honest, gene technology has changed so much since I was at university. It's probably changed a hell of a lot since they actually created this specification. So you don't need to worry about talking about the sequence or how the chromatin condenses or decondenses. None of that's in here. So to summarize, you have a target gene that is useful somewhere in the DNA, which lives inside the nucleus. Either side of that DNA, of that gene, you've got regular DNA. Regularly throughout the DNA, there are these palindromic sequences that read the same forwards as backwards. Sometimes they're just a couple of bases long. Sometimes they're a little bit longer. They're not generally that giant. So they just read the same forwards and backwards in the same section. And there are enzymes called restriction enzymes that have an active site that is complementary to be able to cut the DNA at those specific sites. The sequence of the palindrome determines which enzyme is specific to it or not. Why do they cut like that? It's just the mechanism by which, I'm guessing, the orientation that it holds the DNA in whilst it's forming the enzyme substrate complex is going to break two phosphodiester bonds, and it's going to break the one at the beginning of the sequence at this, because it can only basically break, if I were to zoom in here, I think this will make sense. And just wait for the. So if this is our DNA strand here, then it can only break down this bond between the C and the G. Oh no. Maybe, I guess it's after the TC. You see here we've got CT and here we've got CT. So it's actually basically breaking the DNA at the same place, but on opposite strands. And because it's on opposite strands, it's indifferent. It's not a clean cut, but it can only break it at the end of that or the beginning of that palindrome, depending on which way you're looking at it, because they're the same forwards as backwards. Does that kind of make sense? It's breaking it, if you're reading it from the G to the C end, it's breaking it at the same place on both strands. It's just that they don't line up because DNA is anti-parallel. Okay. Everyone vaguely, any more questions on that? I don't want to move on too much until that's been done. Yeah, I mean, you're quite right, Matilda. It is, if this is your first exposure, then using the course videos, obviously they'll be going at more at your own pace. It's a restriction enzyme, not a restrictive enzyme. It's just the name of that class. You can also call them restriction endonucleases if you want. Sometimes teachers will call them that. That's totally fine as well. Yeah. I'd say that's a great definition, Nesi. Enzymes that cut DNA at specific palindromic sites or specific recognition sites. And they're obviously going to break the phosphodiester bond. That's sort of a common link back to the structure of DNA. I'd say the second one that you're most likely to need to know, I'm probably not sharing my screen right now, so it's not going to be that useful. And it's using reverse transcriptase. Okay, so cells only have 2 copies of each gene, which are in the nucleus, which makes them difficult to access. Reverse transcriptase is an enzyme that does transcription backwards. Okay, so ordinarily, you'd go from regular DNA in the nucleus, and we'd make messenger RNA. What enzyme does this normally in this direction? What are we making? It's a polymerase, and we're making a polymer. But we're going from DNA to what? We're making RNA. So it's the polymer that makes RNA, which means the enzyme is RNA polymerase. So whatever it's making is the letters at the front, and then the polymer making a polymer. Ase is the enzyme. So this is the enzyme that makes the polymer of RNA. And so in this specific instance, oh, maybe I'll do this in a different color. We're going to go from messenger RNA backwards into DNA. We call this type of DNA, someone's already raised this in the chat already, complementary DNA. I'm going to label this first, and the enzyme here is reverse transcriptase. And this DNA is called complementary DNA. Which contains no introns. Because viral DNA or messenger RNA does not contain introns. There's no introns. They are removed to make the messenger RNA. So this is useful because there's loads more copies of messenger RNA. Maybe I can add that. Obviously, there's many more copies of messenger RNA if that cell is producing the protein that you're looking for. If you go to an eyeball cell, you go to a cell in the retina, a rod cell, it's not going to be expressing insulin. So there won't be messenger RNA for insulin just floating around in the cytoplasm of your rod cells in your eye, or the axon of a neuron, or any given cell. But if that cell is producing the protein that you want, then there will be loads of messenger RNA available. That's not called splicing in that reverse direction. No. Splicing would happen only during transcription. the forwards direction. Technically, RNA polymerase would make pre-messenger RNA or primary messenger RNA, primary if you're OCR students, or pre-messenger RNA if you're anybody else. And then that would undergo splicing to form mature messenger RNA or mRNA. Yeah, I mean, in general, in terms of using reverse transcriptase, it does the full process would be faster than by going into the nucleus and extracting the DNA. cDNA is complementary DNA. It's the DNA that is produced using reverse transcriptase. When you start with messenger RNA, you run it, you basically convert the messenger RNA into single-stranded, which is another thing which I did not put on my notes there. It's also single-stranded. So you can add that just below the no introns and single-stranded. You can obviously then make it into a double-stranded if you need it to. Shall I try and find a simple question? I think maybe I'll run you through a question, and I don't know whether, and then I can answer any questions you've got about what your teachers are giving you and any help that we can give outside of these live sessions here. I'm gonna open all three of these documents actually, because I don't know. I've got one question in mind, but it's an AQA question. I'd like to try and find an EDD Excel example, just to, because EDD Excel students always tell me that okay. It's going to be right down the bottom so I can hide. EDD Excel students quite will often say, oh, I don't, my teacher doesn't cover this. This is not part of my my specification. Okay, this is an EDD Excel new spec question. It's right down the bottom here. So I don't know what tabor is. They used enzymes to genetically modify a harmless species of E. coli. It's from the scientific article, so it's a huge block of text, but basically they're using enzymes to modify a species of E. coli, a bacteria. Describe the functions of the enzymes used to genetically modify bacteria. Okay, so we're going to have to name the enzymes. We're also going to have to then say what function that they do as well. So it's a little bit inconvenient. In fact, what I can do is, yeah, I can't scroll it up because the answers are just below. Okay, so don't just give me it, like, give me enzyme and function, because that's going to be what you need to do. We're describing the functions is what the question says there, look. So we really need the function to get the mark. Ideally here, we're going to use 4 enzymes. It might not be that you can think of 4 enzymes, and that's okay, because you might actually get a mark for naming it. You might get a mark for describing its function. Okay, so restriction enzymes, fragmentation cuts DNA at specific recognition sites. Yep. To cut out the target gene. Or you can use reverse transcriptase to convert messenger RNA into complementary DNA. I think that's a nice one. I haven't gone into the inserting target genes in this point. This is an interesting question. What do you use when you've got your sticky ends and you then whack it into a new piece of DNA? You can use the same restriction enzyme to cut open the same flaps, basically. So your sticky ends are complementary. How would you seal up? It's naturally going to form some hydrogen bonds. How would you seal up that DNA? This is just a question. Well, I haven't covered this with you yet today. Matilda, DNA ligase. So Matilda, can you tell me the difference between DNA ligase and DNA polymerase? Okay, I'll scroll down so we can see the answers. Notice how sticky ends is always in here, always. That's a key term. YouTube, come back to me. So DNA ligase is the bonds between the backbone. Okay, so this is it forms the same bond. DNA ligase and DNA polymerase both form the phosphodiester bond between nucleotides in the sugar phosphate backbone of DNA. So both form the same bond, but the difference is DNA ligase is going to take two long molecules, two polymers. So imagine two long trains and like joining those trains together. It's also going to do it for double-stranded DNA. So you're going to join two long things together. DNA polymerase is good. You've got the one long train and you're going to add on one nucleotide, one carriage at a time. And you're just going to add on one carriage and then move on, add on one nucleotide, add on one nucleotide. You're going to be building the polymer. Whereas DNA ligase is going to take the two long pieces and then hook them up together to form the continuous strand. It's like just like meshing them together. Does that kind of make sense? It's not a common, because students will often use them when they are not interchangeable. If you're trying to merge two pre-existing large pieces of DNA or hundreds of bases or more, then you'll use DNA ligase. If you just want to add on a single nucleotide at a time, then you'll be using DNA polymerase. Okay. I'm going to come back to this topic tomorrow. We'll do some more about inserting them, so more about using DNA ligase and a bit of marker genes as well. DNA polymerase adds one base at a time. Just think that's your classic DNA semi-conservative replication, uses DNA polymerase, and obviously during that DNA synthesis, you're adding one nucleotide at a time. You only really come across DNA ligase here in gene technology. It does rear its head a little bit if your teacher goes into the, in fact, I'm not even going to raise it now because it's just going to, if you get Osaki fragments, so sometimes Okazaki fragments, if sometimes OCR teachers will go into that much detail, you've then got two double-stranded, you've got two pieces that need joining together. Okay, so yeah, Edith, are you a subscriber? Do you have the course? Yeah, so because DNA is anti-parallel, on one strand you can form DNA continuously during DNA replication, and on the other strand it can't, because of the way the replication bubble works. It can only do, the DNA isn't unzipped far enough, so it can just do a little bit and it has to detach and then go back and do a little bit more, and then you end up with all these fragments. They're called Okazaki fragments, and then they have to be joined together, and it's DNA ligase that does that. Again, you've got these two long lengths of DNA, not just adding one at a time. Okay, what exam board are you, Edith? There's 2 sessions scheduled for Gene Tech, although it's actually quite a chunky topic depending on your exam board. You're at OCR, okay, so there's plenty in there in the OCR version of the course for you. So I would say run through the videos. I'll be doing the sort of inserting target genes, maybe some marker gene stuff, because that is probably most universal to everybody. What's the OCR heavy part of this? There is a giant old spec question on this, which is basically tell me everything you need to know about gene technology for 9 marks, which I might bring up just to exemplify all the different angles of it. Basically, it's an OCR question. In the beginning, it will let you give you the choice. You don't have to go into the methods we've talked about today. You would say either use restriction enzymes to cut out the gene, or you would say use a reverse transcriptase to form complementary DNA from the messenger RNA. It's a tell me everything you know. It's one of those ones that if you know those key points, and again, those red words I put on my notes there, restriction sites, restriction enzyme, forming sticky ends, like these are the words that are going to be in the mark scheme on those questions. Yes, I can do some predictions. We were planning predictions for the May-June exams. I'm going to tell you right now, no matter how much research I put into it, I can't predict with 100% accuracy. The only way to be completely confident going into those exams is to know all of the content. But even if you know all of the content, the devil in A-level biology is the exam technique. So you've got to basically understand most of the content fairly well. You don't have to know it all perfectly. There are still bits that I forget sometimes. So it's like, what's the difference between a cofactor and a coenzyme? It's like, which one's organic and which one's inorganic? And like some bits just slip out your head, and that's going to happen. But getting the exam technique polished is, you're going to need to basically have the content in your head when you get your calculated grade, so that you can then just fine tune the exams. So just relying on a prediction is I can help. I'll do everything I can for those people, because there will be thousands and thousands of students who are not that well prepared for the exam and will want to take an exam to prove themselves. I will do what I can, but the only real way is to learn it relative. It's not an exam that you can just like, oh, I'll just blag it, because it's not really a blaggable exam. We have the AQA essay is 25 marks, so count yourself lucky. Yeah, so what are your teachers doing with you? I've got 5 minutes max, because Ronnie's going to need the room to start hers at midday. What inside the course for our subscribers, I'm going to be doing some additional live classes this week. We were just discussing that this morning with Rich and Ronnie. We're going to do some drop-in Q&A type stuff, so you can bring us questions and we can go through things like that with you. I've got to do one more live class on the practical skills for my students, because we basically hijacked that session 2 weeks ago to get some, it was just after they announced the exam cancellation. So we just wanted to see what you students wanted, really. The teachers started using Loom, so they just pre-record little bits and pieces and then send it out to you guys. The exams are going to take place if you are not happy with the grade that you're given. You'll be given a calculated grade. If you don't like it, you can do an exam. How long are the sessions that she records on Loom? Microsoft Teams. Still nothing on private candidates as far as we can make out. We actually have some information that you would like to know, but we can't tell you yet because it has to be announced publicly. We found out through our contacts, and don't ask me, because I will not tell you until we can. As soon as we can, we will release a video on YouTube and we'll do a YouTube live. It's about how your grades are going to be awarded. Well, Edith, you've got the course. I mean, obviously, using the TT method, have you more or less completed the content? You said you haven't done the gene tech yet, but that's towards the end of your units component 6 for OCR. So I'd say use the content guide to complete your notes on all of the content, and then use the TT method just to keep fresh on, do 1 paper a week, 1 cycle of the TT method per week. And we're going to be doing more of that. And I'm going to be doing some more live classes with you. So if you've got any specific questions, you can ask me, obviously in the comment below any video, but I'm going to be a bit more available to do drop-in Q&As within the course for you students. And maybe some extra live classes on some exam technique and stuff like that. So you gave me all your feedback on what you wanted to do in these revision live classes. Obviously, everything's been a little bit ripped up and thrown up in the air, but we're still going to be there for obviously all our subscribers. Okay, Amy, great. Okay, well, I mean, obviously I'm only here 45 minutes a day, which may or may not be enough in terms of completing the content. I'm also having to restrict the content to content that's universal to nearly everyone, which means that I can't dive. Normally, the way that I teach is to go super specific into what your exam would want you to answer for those exam questions. That's just my style. I know how I can support you best in there. It's just quite difficult to do that on YouTube because everyone's doing different things. I don't know. To be honest, how many people we got here right now? Like, we're gonna just keep reacting. It's like only 30 of you here right now, so we're just gonna review what to do with the teaching method. Maybe we'll do a couple of sessions so you can get you set up so that you know what to do and when with the live classes. Everything's we're just winging it and making it up as we go along right now. We wanted to basically do some content live classes to make sure you've got a good handle on the rest of the content. Obviously, you can do the TT method flexible. You can do it to your own schedule. You can do as much as you want. You can do as little as you want. I might do some timed papers, some marking, maybe some suggestions. But if we're only getting like 30 or so people, then I'll just do it inside the course for our subscribers. It seems like maybe YouTube isn't the best spot for it. Well, there's only 6 hours in the day, and to be honest, if it's too early, then that's their loss. Well, thank you all for joining me. On which note, I've got 3 minutes. I need to jump out of this room before Ronnie kills me. Okay, see you all tomorrow.